Assalamu alaikum. Assalamu alaikum.
Hello, am I audible?
Hello. Sir, assalamu alaikum.
Walaikum assalam. How are you?
Fine, and you?
I'm also fine by the grace of Almighty Allah. So here we have two participants. One is, I think, I am myself, and the other one is Sayyid Awais Shah.
Yes, sir. All class below present, sir, before. We can leave the site
Why?
now. Sir, present नहीं है ना.
Okay, no.
टेम मालूम. ओके, टेम मालूम नहीं, लेकिन सर टाइम टेबल जो है वो तब्दील हो चुका है, क्योंकि 9:30 से अभी 12:30 तक है, लेकिन पता नहीं सर किस टाइम क्लास लेता है, तो उन्होंने साइट को लीव किया हुआ है. हम 10 मिनट पास वापस आते हैं.
Okay, Sadiqullah is here, I think. Okay, inform your friends to come and join the class.
ओके.
Text them in the group.
ग्रुप में मैं ऐड नहीं हूँ सर, मैं न्यू हूँ, मैं अभी फ्रेश हूँ, फर्स्ट सेमेस्टर है. ग्रुप में ऐड नहीं हूँ, लेकिन मुझे
Okay, I
किसी के
think
नंबर मेरे पास नहीं है. ओके.
Sadiqullah is the admin of that group, so I'll tell him to add you. Sayyid Awais
यस
Shah,
सर.
right? He
यस.
will add you, inshallah. Okay, Maryam Ghaffar is here. Can you hear me, Maryam?
Assalamualaikum, sir.
Walaikum assalam. You are a student of which semester? First or second?
Second semester.
Second semester. Okay. Stand line. Okay.
Okay.
Okay, Sayyid Awais Shah, do you have contact numbers of your friends?
जी सर, मैंने शेयर किया है सर, मैंने इस टेक्स्ट में मैसेज किया है सर, टेक्स्ट
Okay.
किया है मैंने सर, शेयर किया है.
Okay, it's okay. So let's wait for them to join the class, and after 5 minutes we will start. Class, if a class is going to start from 9:30 and it end at 12:30, it means that all of you must be present at 9:30. You
ओके
know,
सर. ओके.
so yes,
उन्होंने
yes, so all of you are supposed to be present at 9:30 till at least 10 o'clock, and then keep checking, because any teacher, he can get late by 5 minutes or maximum 10 or 15 minutes. So
ओके.
30 minutes mean that the class will start exactly at 9:30, or with a
ओके सर.
fluctuation of time, of 10 or 15 minutes. Okay. Sayyid Awais Shah, if it is your first semester,
Yes
it
sir.
means you are a bit away from selecting research topic. But what was your research topic in MPhil?
My research topics are introduced.
Yes. What was your research topic?
Okay sir. My name is Sayyid Aisha. I have done my MPhil from Portable University, Peshawar, in 2021, January 2021. My research topic is ethno-eternal study of important plant from selected localities of the Himalaya.
From selected localities of?
Ethno-eternal study of important plant from selected locality of Takhtabai.
Okay, Tahbai. You
नेट
have,
प्रॉब्लम है सर यहाँ पे
it's okay, it's okay.
तख्ताबाई से
You are audible. You are audible. Takhtabai. So you have selected some specific areas in Takhtabai and you have selected plants from over there.
localities. Yes sir.
Which localities? Which specific localities?
एक यूनियन कौंसल टक्कर, एक मरीबा, एक जलाला, ये 3 मेन यूनियन कौंसल है। वहाँ पे मैंने इन 3 यूनियन कौंसल पे काम किया था इतने टर्म में।
So you have reported some floras of those areas.
Yes sir, 78
Vegetative.
plant I have collected, 78 plant.
Okay, that's great. That's really a great work. So what do you think? Anyone, is there any plant which has not been reported so far for different kind of pharmacological activities?
जी सर। सर
Uh,
नेट प्रॉब्लम है, आवाज मिस हो रहा है।
it's okay. It's okay. Sayyid Awais Shah.
जी
So,
सर।
okay, can you please name some plants which are important and which are present only there
वेयर सर? यस सर,
in these areas?
Morus alba, Morus negra, Eltesimia brassica campestris, Pinus raxburgii, Pinus gerardiana,
Okay, Pinax roxburgii is there.
Cucamus melia zadarecho, Tunica granitum. Lots of I have collected 78 plants, which is
Okay.
which help in 41 different diseases of veterinary.
Okay, that's really great. Sayyid Awais Shah, you can help your friends, your colleagues, and selecting plants for their research because you have already worked on plants which are available there, and you might be having an extensive review, literature review of all these plants, so you can help your friends. All the
Okay, okay,
all the ethnobotanists, they are assets of botany. So
okay.
they
Why not,
can,
sir?
yeah, so they can tell us about the plants, about the novel plants and about the rare plants which has not been investigated so far. So you can help out your friends. So it's a good field. Will you
Okay,
continue ethnobotany or will you go for some lab work?
sir. Inshallah, I have taken related to that topic in PhD. Inshallah, I promote that topic in PhD.
Okay. Okay, that's really great. Okay, Mariam Wafar, tell us about your research.
Lab work, where the lab work, inshallah lab work भी अच्छा है, लेकिन फील्ड वर्क भी अच्छा है सर। लेकिन मेरे पास कुर्तुबा यूनिवर्सिटी में जो है लैब वर्क काफी मुश्किल तो नहीं होता, लेकिन सामान जो है
Okay,
से
so
हमें उन्होंने हमें तो कहा था, लेकिन हम आपके लिए सामान जो है वो बराबर करेंगे इंशाल्लाह। अगर आप यहाँ पे काम करते हैं लैब में,
just
तो हम आपके लिए
go
इंतजाम
for,
बराबर
okay,
करेंगे।
Sayyid, always go for whatever you want to do, as per your desired design of your research. You can go for that. We do have lots of research centers and we can work there being scholars. We
ओके
can
सर।
work in all the research centers which are present in Peshawar, specifically in KPK. In general, right? So
यस सर।
just design your work the way you want, and then we will see in which specific research centers you can work, right? Just do it and go for that. Okay, Sayyid Awais, thank you for all the information
ओके
which you shared with us. Now I would ask
ओके सर।
Mariam Ghaffar to tell us something about her previous work in MPhil. Okay, Mariam, can you hear me?
Sir, I done MPhil from University of Peshawar.
University
मसला है,
from University of
लेकिन
Peshawar
फिर
you
भी
did
अगर
your,
मेरी आवाज आ रही है ना तो
okay, you
मैंने
are,
MPhil
go ahead, you are audible. You are audible.
हेलो
Okay, you are audible, Mariam. You are audible. Go ahead. You
My
did
research
masters.
topic was ethnobotany.
Okay.
University of Peshawar.
Okay, that's great. Who was your supervisor?
Supervisor.
Yes, and MPhil, who was your supervisor?
Dr.
Dr. Mukarram Shah. You have done your MPhil from which? From biodiversity?
Sir, आवाज clear नहीं आ रही मुझे.
I asked that you have done your MPhil from biodiversity.
Sir, आवाज clear नहीं.
Assalamu alaikum, sir.
Wa alaikum assalam. Amir Sultan is here.
Sorry.
Yes, sir. How are you, sir?
I'm fine by the grace of Almighty Allah. What about you?
Thank you, sir. Thank you, sir.
Why are you late, man?
Sir, I was the first to join the meeting, but sir, no one was there, so I leave there.
Whenever I open this chat and this meeting, the one who can I see is Amir Sultan, and today Amir Sultan was absent, so that's why I missed him.
Sir, I was logged in proper in 9:30,
Yeah.
but there was Hatak Saab and Atifa Kudus and Aves Shah. We, for a while, we talked to each other, but we thought that sir will probably join us at 10 or 11 o'clock. That's why,
Hmm.
sir.
Okay. So I told Sayyid Awais Shah and Mariam that you must be there. When you join your
Sir,
class, you can discuss things.
yes, sir. I have given the text to them on the WhatsApp group, sir. Now,
Okay. That's good. Murad Ali Khan has also joined us.
sir, they will join us now. Aves Shah is
Okay.
also here.
Okay. Murad is here. And I
Yes.
think Sadiqullah is missing and Atifa is missing presently.
Yes, sir.
Okay, I was talking to Mariam. Mariam, can you please tell me which specific department you have done MPhil in? Botany or biodiversity?
PS botany में है और MPhil biodiversity में.
Okay, MPhil biodiversity. It's okay because you said that your supervisor was Sayyid Mukarram Shah.
Yes.
And your research title was?
Sir, weeds पे था. Different weeds.
Different weeds from where? From Peshawar or somewhere else?
नहीं Malakand, from Malakand area.
So you have worked on weeds. Which kind of work? Have you elaborated some medicinal value of this plants like pharmacognosy, pharmacology, or which specific activity? Mariam, can you hear me? I think this will not be an impersonation case because I have been impersonation officer for some time, so it's okay. We'll talk over it later. Atifa, you joined us. Atifa Quddus is here.
Yes, sir, I'm here.
Sir, sorry. ये नेट का कुछ इशू हो रहा है ना आवाज.
Okay, you are audible now, Mariam.
I'm here, sir.
Okay, let's start. we can discuss things later.
Senior.
Let's start our lecture. In the name of Allah, the most gracious and the most merciful. We have discussed recombinant DNA technology in detail, and we have also discussed all the enzymes present in recombinant modification system. There were 4 systems of recombinant DNA technology, which is known as recombinant modification system, RMS. And there were 4 types of RMS, in which RMS2 was the most commonly used system. Recombinant modification, restriction modification system 2 was the most commonly used system, and which even we used for isolation of DNA, for preparing, for manufacturing, for engineering recombinant DNA. Recombinant DNA, as you can see on your screen, is that DNA which has been manufactured or engineered artificially, which is not present before. Either there are 2 choices, there are 2 options, there are 2 ways. The first one is that it is totally not present, what we want to prepare or engineer. And second is it is present but not an appropriate amount so that we can get it. For that specific purpose, we manufacture DNA in 2 different medias. What are these 2 medias? Can any one of you tell me? I think except 1 or 2, all of you are present in our previous lecture. So will you tell me that which are the 2 medias wherein we can engineer recombinant DNA?
Sir, PCR.
Yes, exactly. We will talk about all those. That's great. What are the 2 medias? Generally, we can name them. Or I would say culture, so it will become more easy for you to identify my questions or to recognize it. What are the 2 cultures of this recombinant DNA technologies, and which 2 specific cultures we can manufacture it? Or I would say amplify it. Okay, let me tell you, this meeting will end in 10 minutes and then I'll start it again, right? So you, all of you will stay with me. You will not go anywhere.
Okay, sir.
Okay, now tell me, what are the 2 cultures? Murad Ali, can you tell me about the cultures which we have studied, wherein we can manufacture recombinant DNA, or we can engineer it, or we can say amplify it? Atifa, will you tell us what are the 2 medias, or we can say cultures?
सर ये जो टिश्यू कल्चर करते हैं हम,
I said I use the word media because it will become easy for you. Which are the localities, I would say, which are the places where we can get recombinant DNA? My question is clear to all of you or not?
Sir, maximum Gilbert process PCR.
These
Polymerase
are
injection, sir.
actually steps. No? These are the steps, and that is one of the step. So a step is another phenomena. And I asked you that which are the areas where we can manufacture or engineer or amplify this recombinant DNA. So what are those areas and how much are they? Where can we manufacture recombinant DNA?
सर ये जो माउस वगैरह के सेल्स में कल्चर करते हैं उसको,
This is a kind of answer because you name cell and a living cell. Then we will come toward it.
one is vivo and one is vitro.
What? That's the answer, right? So before asking questions, you take me there towards the answers.
Yeah.
Okay. Now, who will tell me clearly what are the areas where we can manufacture or engineer recombinant DNA? What are the modes? Modes, you understand now?
The process, sir, the procedures.
Okay, that's great. Uh, Let me tell you, the first one, the first mode or area, that is test tube. Is it clear now?
Yes, sir.
Okay, so as I give you answer, now my question is clear. So whenever I will ask you questions, before that I will have to give you answers as well. Then it will be easy for you to understand the question and then you will answer the questions. Is that so?
No, sir. Maybe not in future.
Okay, that's great. Shireen Zada, are you here?
Yes, sir. Assalamu alaikum, sir.
Walaikum assalam. Where were you, man?
Sir, I'm sleeping, sir.
What? You are late?
Sorry, sir. I'm a little bit late.
Okay, I asked your colleagues that what are the areas, what are the modes, and what are the media where we can manufacture or engineer recombinant DNA. So what would you say about it?
What are the areas here?
Amir Sultan, tell us. As you have been listening all these questions, so you have got the answers, I think. And now tell us those areas where we can manufacture our engineers the nexus.
Recombinant.
Sir, recombinant DNA technology, sir, we use the process is that gene of interest, they are selected, and we are having biological scissor for that.
That's
And to
great.
insert it to a plasmid, proper medium, sir, that is plasmid PBR 222, that is used mostly for that process.
Okay.
Okay, sir.
Sir, then we have a culture bacteriophage use that is used mostly for that purpose, sir.
Okay, that's really great. So which two?
Scientists, sir, they have further worked in the PCR machine, sir. These are
That's
some of the
great.
processes.
So which are the two specific areas where we can work, or two specific cultures? There are two cultures which we can talk about while
Yes, sir.
thinking of making recombinant DNA. So what are those two areas, or what are those two cultures, or
And which was, sir?
Okay, that's great. One is test tube and the other is living
Yes,
cell, right?
Yes,
So
sir. And view and which, sir.
exactly. So what we do, we actually we get a piece of segment in a test tube, and
Yes, sir.
then we ligate it with another same sequence of base pairs to that piece of which we have been selected our targeted gene. Right after ligations, we transform it to the living cells where they replicate its numbers, and then we obtained that DNA which has been engineered, right, complementary DNA, and we get that and we utilize that for the purpose which has been engineered, right? It is clear now.
Yes, sir.
So the first one is
Yes, sir.
artificial and the second one is a living cell, and that's what I wanted to ask you. So is it clear to all of you?
Yes, sir.
Yes, sir.
Okay, now let's come toward this topic. We have discussed in detail the process in which a recombinant DNA can be obtained or engineered, right? And we have also talked in our previous lectures about the enzymes which are used in this very specific method or technology, right? So I told you that restriction modification system 2 is widely used because that is quite accessible, and we can have two types of enzyme. The one is restriction endonuclease and the other is modification enzyme, right? So methylase, and we have also read about all the enzymes which are used in all these four systems, right? We can follow one of the system in getting recombinant DNA. There are actually four such systems. So system 2 is widely used, and we will talk about system 2. And here I will tell you that what is this technology, and we will have a look over the whole procedure. This is a kind of summary. So attending this lecture, you will understand that what is recombinant DNA technology, and you will come to know about all the steps in a brief form, that what actually recombinant DNA is and how can we carried out this activity, and how can be recombinant DNA obtained, and this can be amplified, right? So this is a general we can say overview of what we have been talking in previous lectures. In previous lecture we were a bit specific, and we have talked about all the enzymes. Not all those enzymes were specific to all the restriction modification system. Like not all those enzymes which I think they were 10 in number, and not the whole of them they are used in one, like restriction modification system 1 and restriction modification system 1 out of 10, some of the enzyme will work, right? And then system 2 out of 10, some will work. So as a whole they were 10, I think they were 10 in numbers, and they were specific to different restriction modification systems, right? So let's talk about restriction modification system. And here on the screen you can see recombinant DNA technology. So you are very much clear about it now, that this is a kind of technique or procedure, prescribed procedure, in which DNA